Dear students, today I am with the third chapter of class 12th biology, that is human reproduction. In this PPT, I am just going to deal with the male as well as the female reproductive system and the processes that take place in them, such as spermatogenesis and oogenesis. Let us start with the male reproductive system. Human male reproductive system consists of primary sex organs, secondary sex organs, as well as the external genitalia. The main organ is a pair of testes. Then the secondary sex organs involves the ducts as well as the glands. Then finally the external genitalia. Here you can see in sequence the parts of the male reproductive system: a pair of testes, then the retitestes, vas deferentia, epididymis, vas deferens, seminal vesicles, prostate gland, bulbourethral gland. These are also called the Cowper's glands, and finally the penis. So the spermatozoas are formed in the testes. From the testes, they are transported to the vas efferentia through the retitestis. Then from vas efferentia, they are transported to epididymis. Epididymis leads to the vas deferens, and in the vas deferens, the seminal vesicles release their fluid known as the seminal fluid. Prostate glands release the alkaline fluid. Bulbourethral glands releases the mucus, and finally all these are being ejaculated out to the external genitalia. This is the internal structure of the male reproductive system, where you can see a pair of testes over there. Each testis, now see, the testes are located outside the abdominal cavity. They are present inside the scrotum, and the scrotum provides 2 to 3 degree less temperature than the body temperature for the differentiation of the spermatozoa. Each testis, now it consists of 200 to 300 testicular lobules, and each lobule consists of seminiferous tubules. These seminiferous tubules are the sites for the synthesis of the spermatozoa. The spermatozoa which are being formed inside these seminiferous tubules, they are transported to the vasa deferentia by means of a duct system known as the retitestis. From vasa deferentia, they move to the epididymis, and epididymis is the site where the spermatozoa are stored, and the maturation of the spermatozoa also occur. From epididymis, the spermatozoa are being transported to the abdominal cavity through the vas deferens. Vas deferens opens into the urethra. Urethra is the opening of the urine. This is the urinary bladder. The opening of the urinary bladder is the urethra. In males, the urethra is being extended and into a structure known as the penis. While when the vas deferens, they just pave their way towards the urethra. Here the seminal vesicle, a pair of seminal vesicles, they open into it, and these seminal vesicles release a fluid known as the seminal fluid. The seminal fluid is rich in nutrients such as fructose and other things. These nutrients, they nourishes the sperms on the way to the urethra. In fact, in the urethra here you can see the two flap-like structures. These two flap-like structures are the prostate glands, and they release the alkaline fluid in the urethra to neutralize the acidity caused due to the urine. And in this way the sperms are being protected. Just below it you can see again a pair of glands which are called the bulbourethral gland or the Cowper's gland. This releases the mucus which helps in the lubrication of the penis during the coitus. If you see the section of the testis, what we see, the seminiferous tubules. You can see the seminiferous tubules here. These are the seminiferous tubules here, and these seminiferous tubules, they are being lined by the germinal epithelium. Now they consist of the two type of cells. Number one, the germ cells, male germ cells, and the second comes the Sertoli cells. These Sertoli cells, they provide the nourishment to the developing spermatozoa. In between the seminiferous tubules, these are the cells here, interstitial. They are called interstitial cells. Also we call them Leydig cells. These cells, they release the male hormone known as the testosterone. Now this is the flow chart which is showing the spermatogenesis. Spermatogenesis, again I am repeating, that is the formation of the sperms in the testis. Now how it is happening? Please see inside the seminiferous tubule. I have told you they are the male germ cells as well as the Sertoli cells. So talking about the spermatogonial cells, these male germ cells, they redivide by means of mitosis, and they are called the spermatogonias. These spermatogonias, after undergoing repeat mitotic division, they stop dividing by mitosis and are ready to move into meiotic division, first meiosis, and at that time they are known as the primary spermatocyte. It means the ploidy now. If it is being asked of the primary spermatocytes, then they are diploid. These are, again you can say, the sperm mother cells also, you can say. These primary spermatocytes, they undergo first meiotic division. As a result, two cells, you can say, the daughter cells are being formed. These two daughter cells, now they are called the here secondary spermatocyte, and here you can see the ploidy. 23 chromosomes have been written, so they are haploid. These secondary spermatocytes, now they undergo second meiotic division, resulting in the formation of non-motile structures here, which are called the spermatids. These spermatids later on differentiate and they develop the flagellum, and they are called the spermatozoa. So you just remember the differentiation of the spermatids into spermatozoa is known as, there's another term for that, spermiogenesis. So spermatogenesis, whatever I have just told you, it has been mentioned over here. At last, when I said to you about the spermiogenesis, what happens to these spermatozoas? These spermatozoas head, or the sperm heads that get embedded inside the Sertoli cell, which I have just told you here, Sertoli cells, and then later on they are released from the seminiferous tubules. We call this process as spermiation. So please do not get confused with the terms spermiation, spermiogenesis, as well as the spermatogenesis. Now the spermatozoas have been formed. Let us see the structure of each spermatozoa or spermatozoon or the sperm. This is the mammalian sperm. The diagram is generally being asked in the exam. Many times has been repeated. So each sperm or spermatozoon, it consists of the three regions: the head, neck, and the tail. The head, now it's the main part because it consists of the nucleus, the tip of the head. It is a beak-like structure which is known as the acrosome. This region, it is rich in various enzymes, and these enzymes are known as sperm lysins. They help in penetration of the acrosome or the sperm into the female gamete, in fact. So in the middle piece, you can say here, this portion, it is rich in mitochondria. These mitochondria, they are called the powerhouse of the cell. So they provide energy for the movement of the flagellum, so that the spermatozoa or the spermatozoon or the sperms, they can move up to the fallopian tube for the fertilization. Now besides this, there are certain hormones also which regulate the process of this which regulate the male reproductive system or the spermatogenesis. So you can see in this flowchart it's being written GnRH. RH means the releasing hormone and Gn means gonadotropin. Gonads means the reproductive organs. So these releasing hormones are released from the hypothalamus, which is present in our brain. Now this hypothalamus, during the puberty time, the hypothalamus guides the pituitary gland to release its hormones. It releases the GnRH hormone, gonadotropin releasing hormone. Then pituitary gland, it releases two hormones from it. They are the luteinizing hormone as well as the follicle stimulating hormone. Remember, this luteinizing hormone, it acts on the Leydig cells and releases the testosterone, which helps in spermatogenesis. Whereas FSH, it acts on the Sertoli cells. Sertoli cells, they help in you can say nourishing the developing spermatozoa as also help in the process known as the spermiogenesis. Spermiogenesis, I'm again repeating this process, such like the differentiation of spermatids into spermatozoa. Hormonal control, the same has been explained over here. Now let us move to the female reproductive system. Female reproductive system now, it consists of the primary sex organ, secondary sex organs, external genitalia as well as the mammary glands. Now primary sex organs means here a pair of ovaries. Secondary sex organ consists of the duct system and the fallopian tube, the uterus, cervix, the vagina, and the external genitalia, then finally the mammary glands. This is the diagrammatic view of the female reproductive system, which consists of a pair of ovaries, then the fallopian tube, and the sac-like structure, which is called the uterus. Uterus, uterus now, the mouth of the uterus is called the cervix, and here there is a cervical canal, or we call it a vaginal canal, also the birth canal, and this is the exterior opening, which we call it a vagina. Now the inner lining, see, you can say the uterus, it consists of the three layers. The outermost layer is called the perimetrium, the middle one is called the myometrium, whereas the innermost layer here, you can see the finger-like projection also. It means thickening is there, not finger-like projection, you can say thick wall, we call it a endometrium. This is the place where the implantation occurs. Implantation means the blastocysts get attached. This is the whole part here, this uterus is the place where the embryo development occurs. So the role of, you can say, the ovary is to release ovum, is to produce the ovum and to release the ovum. Once the ovum is being released, this ovum is being, or the egg is being captured by this finger-like projections of this fallopian tube. Then the egg or the ovum, it trickles down, it moves in the ampulla isthmus junction, and this is the site where the fertilization occurs. This is the process of oogenesis which takes place in the ovary. Now in the ovary especially, we see again the cells known as the inner lining of the ovary. Now it consists of the cells which are called the oogonial cells. These oogonias, they redivide by the mitosis. Remember, in females, the you can say the sexual reproduction, the process of sexual reproduction, or the formation of this, the or the process of oogenesis starts at the embryonal stage and will remain arrested at some stage of the meiosis. So this oogonia which undergo the mitosis, and later on a time comes they stop dividing. See, it's being written over there, the fetal stage. So they stop dividing by mitosis, and when they stop dividing by mitosis, they are regarded as the primary oocyte. This primary oocyte now, it undergoes meiosis. When it undergoes meiosis, just see here, this at the meiosis it enters the prophase 1, and later stages get arrested over here. The female child is born with large number of the primary oocytes. And you can see the primary oocytes, many oocytes, you can see, they degenerate when she attains she reaches the age of the puberty. Now at the puberty, and the influence of, you can say, the female hormones, this primary oocytes, it completes its division. The first division is being completed, and meiosis is completed, and the secondary oocyte is being formed. This secondary oocyte is being released during ovulation in the females at the time of puberty, when she comes in the menstrual cycle. So secondary oocyte waits for the male gamete during the fertilization, and once the male gamete enters in the female's body, what happens? This secondary oocyte undergoes second meiotic division and forms the ovum. Remember here that the division, you get the daughter cells which are being formed as a meiosis, they are not equal. One is very small, you can say, towards the polar end, so we call it a first polar body. It disintegrates normally. What is the function is still not known. And the second cell, this which is large, which has got a good amount of cytoplasm, it is called the secondary oocyte. Then secondary oocyte again divides. Here you can see the second polar body is being formed, and the other one is the ovum. Always normally is this is the ovum. Come to the structure now of the ovary. If you see the section of the ovary, what you find? You can see the different type of follicles over here. I've told you here that it's being written here: primary follicles, tertiary follicle, Graafian follicle. What are these follicles? I've told you that the primary oocyte, which is diploid, which is being formed when the female is in the embryonar stage. So this primary oocyte, it just develops a, you can say, thickening around it, and this thickening is of granulosa cells. We call this structure, here you can see the primary oocyte with the thickening, known as primary follicle. So primary in the female, what is formed when she is taking birth? Then the primary oocyte is like that. It's not free. It is being bounded by the granulosa cell. We call it a primary follicle. Later on, under this primary follicle, especially, it becomes more and more thick, and so you can see the laying layer is called, the outermost covering is known as the theca. So this is called the secondary follicle. This secondary follicle then again matures, it enlarges, the primary oocyte inside it, it just start growing, and then completes its first meiotic division. It converts into secondary oocyte, and again you can see the more and more thickening develops, and here the primary follicle, it is not being written here, the primary follicle converts into secondary follicle, then secondary follicle converts into the tertiary follicle. In this tertiary follicle, how will I differentiate this? Identify the tertiary follicle in the diagram. You see a cavity here. This cavity is called the antrum. If you see, the antrum has been developed and more layers have been formed, and this is the oocyte over here, secondary oocyte. We call it a tertiary follicle. This tertiary follicle, it matures and into a Graafian follicle. This is the Graafian follicle. This Graafian follicle is now ready to burst, you can say rupture, and when it ruptures, it releases the, here it is being written the ovum. In fact, this is the secondary oocyte which is being released. We call this process as the ovulation. Soon as the ovulation occurs, the secondary oocytes is being given out. This is being trapped by the fimbriae of the fallopian tube, moves to the fallopian tube for the fertilization. The wall, you can say, of this Graafian follicle, it converts into yellow structure which is called the corpus luteum, and this corpus luteum, it acts as an endocrine gland. It releases a hormone known as the progesterone, and which especially helps in pregnancy. Then in case if the fertilization do not occur in the female, what happens? This corpus luteum, it disintegrates into the white structure. We call it a corpus albicans and get degenerated. It is easy to speak that the primary follicle has converted into secondary follicle, then the tertiary follicle and the Graafian. All of these events, they are taking place. You can see the maturation or the growth of the follicle is taking place under the guidance of again the hormones. So there are hormones also, hormonal control also in oogenesis. So in this oogenesis, I have already mentioned to you that how many follicles you can see at the time of puberty. Normally 60,000 to 80,000 of primary follicles are left in each ovary. This is the hormonal control. Here you can see the hypothalamus, it's releasing the gonadotropin releasing hormone. This gonadotropin releasing hormone is working on the anterior pituitary gland. Anterior pituitary gland in turn releases LH as well as FSH. This LH and FSH, both of them, they act on the ovary. Remember, this FSH, follicle stimulating hormone, it helps in the growth of the follicles. Primary follicle is converted into a secondary follicle, into tertiary follicle, and finally into the Graafian follicle. So it stimulates the ovary to release its hormones known as the estrogen, and that helps in the development of the follicles. Whereas LH, especially this LH hormone, it is the hormone which helps in the busting, you can say, or the rupture of the Graafian follicle. So rupturing of the Graafian follicle, releasing of the second oocyte from it, is called the ovulation. So you can say LH is responsible for the ovulation. Ovary, it is being mentioned, it is responsible for estrogen and progesterone, and these two hormones again, they act on the uterus also, the thickening of the uterus and other things. Normally the growth of the uterus also occurs. And now if the amount of the estrogen will be more, again it will be giving the feedback to the hypothalamus. Then either the hypothalamus will stop releasing the GnRH, and if it is required, then the positive feedback will be given. In this way, the female reproductive system is also, as well as the hormonal control, is over. See you